and welcome. If you are settling in for the evening, finding your position, letting the blanket settle around you, letting the sounds and pressures of the day begin to soften at their edges, then you have come to exactly the right place. This is the Science Channel for Sleep, and tonight we are going underwater. Not to the open ocean, not to the deep, dark water of the abyssal plain, but to something altogether different. A place that occupies less than 1% of the ocean floor and yet supports approximately 25% of all known marine species. A place that looks, from a distance, like decorated rock. A place that is, in reality, one of the most densely inhabited and biologically complex ecosystems on the surface of the Earth. Tonight, we are going to explore the hidden world inside coral reefs. Before we begin, if you enjoy this kind of calm, unhurried journey through the wonders of the natural world, if this is the sort of thing you like to fall asleep to or rest quietly alongside on a still evening, it would mean a great deal if you took a moment to like this video and subscribe to the channel. It truly helps more than you might expect. It helps other people find their way here, to this particular kind of quiet. And it helps this channel continue making exactly this kind of content. Content that is not in a hurry, that simply wants to share something worth knowing before you rest. And if you feel like leaving a comment, we would genuinely love to know where in the world you are listening from tonight and what time it is there. It is always interesting discovering where listeners are tuning in from around the world. Someone may be in the late quiet of a Tuesday night in one city, and someone in the early grey of a Wednesday morning in another, and someone in a warm afternoon in a part of the world where the day is still going, all of them here together for a little while in the same unhurried space. Now, let us go underwater. Imagine you are looking down at a coral reef from a boat on a calm tropical morning. The water is clear enough that you can see the reef perhaps 10 or 15 meters below the surface. What you see from up here is a landscape of irregular shapes in shades of brown and pale green and cream. It looks like rock. It looks geological, as though the ocean floor in this place simply happens to have a different texture from the sandy bottom around it. If you did not know what you were looking at, you might not look twice. That is part of what makes coral reefs one of the most quietly extraordinary things on this planet. Their surface appearance gives almost no indication of what lies within. Here is something worth holding before we go any further. The structure of a coral reef, the physical material of which it is built, the calcium carbonate that gives it its weight and shape and geological character, was made by animals, not by geological processes, not by the slow chemistry of mineral deposition, by animals, specifically by animals so small that a single one of them is barely visible to the naked eye. Animals that are not fish, not crustaceans, not any of the creatures that most of us would think of when we think of ocean life. The builders of coral reefs are called coral polyps, and they are related to jellyfish and sea anemones. They are soft-bodied, tentacled, extraordinarily small, and they have been building for hundreds of millions of years the largest biological structures on the surface of the Earth. One of the most common misconceptions about coral reefs is that they are a kind of plant, or perhaps a kind of mineral. The word coral suggests something between a stone and a plant, something passive, something that simply grows in the way that crystals grow, following chemical rules rather than biological ones. This is understandable. For most of human history, scientists themselves believed that corals were plants. It was only in the 18th century that careful observation established what corals actually are: animals belonging to the same broad group as jellyfish, living together in vast colonies that collectively build a structure you can see from space. Another common assumption is that the colorful, textured surfaces visible on a reef are what a reef is, that what you see on the surface is the whole story. It is not. The visible surface of a reef is only the beginning. Beneath it, within it, around it, and in the water above it, there are communities of extraordinary complexity and variety that most visitors to a reef never glimpse. Researchers believe that for every species visible on the surface of a coral reef, there may be 10 or more species living within the reef framework itself, in the tunnels and caves and crevices that make up the interior of the reef structure. The reef is not a flat backdrop for the fish that swim in front of it. It is a three-dimensional world of extraordinary depth and intricacy, and the fish are only a small part of what inhabits it. Tonight, we are going to explore that world from the inside. We will begin with the smallest building block, the coral polyp itself, and gradually expand outward and inward to understand the full complexity of what a reef actually is. We will visit the extraordinary partnership that makes reef building possible. We will look at what happens on the reef at night and at one of the most spectacular biological events on Earth. We will explore the hidden interior of the reef structure, the social networks of reef fish, the predators and grazers and invisible engineers that maintain the reef's extraordinary productivity. We will trace the history of human understanding of reefs and sit quietly with both the challenges they face and the science being done on their behalf. One more thing worth saying at the beginning. Coral reefs are sometimes called the rainforests of the sea, and the comparison is apt not only in terms of biodiversity, but in terms of what they represent. A rainforest, seen from a plane, looks like a green carpet. From within, it is a layered, humming, constantly active world of extraordinary complexity. The same is true of a reef. The view from the surface gives almost no indication of the density and richness of what lies beneath. That density and richness is what tonight is about. We will move slowly. There is no rush. The reef has been there for hundreds of thousands of years and is not going anywhere tonight. Let us begin with the animal at the center of everything, the coral polyp, and discover how something barely visible to the eye has built some of the largest structures in the history of life on Earth. That is where we are going next. At the center of everything on a coral reef, at the foundation of every structure, every ecological relationship, every community of organisms, is an animal so small that you could fit several of them comfortably on the tip of your finger. It is called a coral polyp. A coral polyp is typically a few millimeters to a centimeter in diameter. It is soft-bodied, translucent, and in its basic form, remarkably simple in appearance. A tube of living tissue, closed at one end where it attaches to the surface below, and open at the other where a central mouth is surrounded by a ring of tentacles. Those tentacles are typically arranged in multiples of 6 tentacles or 12. or 24, a pattern that places coral polyps in the order Scleractinia, the stony corals, and connects them to their evolutionary relatives, the sea anemones and jellyfish of the phylum Cnidaria. If you were to look at a coral polyp in isolation, a single individual separate from its colony, you might reasonably mistake it for a tiny sea anemone. The resemblance is close. The tentacles wave in the water in the same gentle way. The central mouth is surrounded by the same radiating symmetry. The tissue has the same translucent quality. The polyp is, in its basic body plan, a very simple animal. What makes the coral polyp extraordinary is not its individual complexity, but what it does. Beneath the soft tissue of its base, the coral polyp secretes calcium carbonate, the same mineral compound that makes up limestone and chalk. It deposits this material in a cup-shaped structure called a calyx, building up the walls of its own skeleton from below. The skeleton is composed of a specific crystalline form of calcium carbonate called aragonite, and its deposition is a continuous process. The polyp adds a tiny fraction of a millimeter to its skeleton each day, each week, each year, building upward and outward in a rhythm that never entirely stops as long as the polyp is alive. The living tissue of a coral is only 1 to 2 millimeters thick. If you were to look at a cross-section of a coral colony, you would see that the vast bulk of its structure, everything below that thin outer layer of living cells, is dead skeleton. Decades, centuries, sometimes millennia of accumulated calcium carbonate built by generations of polyps, each one living briefly on the surface before dying and being incorporated into the foundation for the next generation. A coral colony is not one animal in the way that you or I are one animal. It is a society, thousands to millions of individual polyps, each one genetically identical to the others in the same colony, each one connected to its neighbors by a living tissue network called the choanosarc, through which nutrients and chemical signals can pass. In this sense, a coral colony is simultaneously many, and one, many individual animals sharing resources and coordinating behavior through a network that links them all. The growth forms that different coral species produce are among the most visually striking features of a reef. Some corals grow in massive dome shapes, the brain corals and boulder corals, adding only a millimeter or two per year, but building structures that can reach several meters in diameter and persist for centuries. A porite's coral colony in the Great Barrier Reef or the Indo-Pacific may have been growing continuously for 300, 400, 500 years. Some have been dated to over 500 years old using growth bands visible in cross-section, each band a year of the colony's life, readable like the rings of a tree. Other corals grow in branching forms. The staghorn and elkhorn corals of the genus Acropora, adding up to 20 centimeters per year under ideal conditions, building complex, three-dimensional structures of extraordinary architectural intricacy. A healthy Acropora thicket is one of the most structurally complex habitats in the ocean, a forest of calcium carbonate branches providing shelter, attachment surfaces, and feeding grounds for hundreds of species. The oldest known living coral colonies may be more than 4,000 years old. 4,000 years, growing quietly on a reef that existed when the Egyptian pyramids were new, adding their fraction of a millimeter each day, building a structure that has outlasted every civilization, every empire, every written record of human history. During the day, the polyps of most reef-building corals are largely retracted within their calcium carbonate cups. The coral surface appears textured and still. The living animal is present, but not visible. Its soft tissue withdrawn, its tentacles pulled in, the whole colony looking very much like the decorated rock that a casual observer might take it for. At night, everything changes. As the light fades and the zooplankton, the tiny drifting animals on which coral polyps feed, rise from deeper water toward the surface, the polyps emerge. Each tiny cup that was sealed by day opens, and the tentacles extend into the water. Seen under artificial light at night, a coral colony covered in extended polyps looks like a surface carpeted in tiny pale flowers, each one reaching into the water, its nematocysts loaded and ready, waiting for a zooplankton animal to drift within range of its stinging cells. The nematocysts, stinging cells present in the tentacles of all cnidarians, fire tiny barbed threads into prey when triggered by contact or chemical cues, immobilizing the zooplankton, which is then drawn down into the polyp's central mouth. This is the coral polyp feeding, the same basic process of capture and ingestion that its jellyfish relatives perform in the open ocean, but performed by an animal so small that its prey must be measured in micrometers. The colony that builds a reef the size of a house does so one zooplankton at a time, one fraction of a millimeter of calcium carbonate at a time, one night of feeding at a time across centuries. Researchers believe there is something deeply worth pausing on in this, the way in which an accumulation of extraordinarily small acts performed continuously over an extraordinarily long time produces a structure of extraordinary scale. The coral polyp is not trying to build a reef. It is simply living, feeding, secreting, reproducing. And the reef is what happens when it does this for long enough. But the polyp does not build alone. It has a partner, one of the most important partnerships in all of biology, that provides most of the energy the coral needs to build, to grow, and to sustain the entire ecosystem above it. That partnership is where we are going next. Inside the cells of almost every reef-building coral on Earth, there lives an alga, not growing on the coral surface, not floating in the water nearby, living within the actual cells of the coral tissue, millions of them per square centimeter, photosynthesizing in the sunlight that filters down through clear tropical water and passing the products of that photosynthesis directly to the coral animal that houses them. These algae are called zooxanthellae. They belong to a family of single-celled organisms called Symbiodiniaceae, dinoflagellates, related to the organisms responsible for red tides and bioluminescent bays. Individually, a zooxanthella is so small as to be invisible without a microscope. Collectively, they are the engine of the coral reef. The relationship between coral and zooxanthellae is a form of mutualistic symbiosis, a partnership in which both organisms benefit from the association. It is one of the most intimate and consequential biological partnerships in the ocean, and possibly one of the most important on Earth. Here is how it works. The zooxanthellae, living within coral cells, have access to two things they need for photosynthesis: sunlight, which penetrates the clear, shallow water of tropical reefs, and carbon dioxide, which is produced as a metabolic waste product by the coral animal itself. Using these inputs, the zooxanthellae photosynthesize, converting light energy into chemical energy in the form of sugars and other organic compounds, and then they share. Researchers have found that zooxanthellae pass up to 90% of the sugars they produce through photosynthesis directly to their coral host. 90%. The coral animal receives the vast majority of the energy produced by its internal algae, energy that it uses for tissue maintenance, growth, reproduction, and the secretion of the calcium carbonate skeleton that builds the reef. In return, the coral provides the zooxanthellae with shelter, the protected interior of coral cells, safe from the turbulence and predation of the open water. It provides the carbon dioxide that the algae need for photosynthesis, and it provides the nitrogen and phosphorus compounds, metabolic waste products of the coral's own feeding, that fertilize algal growth. The zooxanthellae are, in effect, farming inside the coral, and the coral is the farm. The colors that make coral reefs so visually extraordinary, the vivid purples, electric blues, deep greens, warm oranges, and dusty pinks, are produced largely by the zooxanthellae. The coral animal itself is essentially transparent. Its tissue is a pale, translucent material that gives very little color of its own. It is the billions of zooxanthellae living within the tissue that provide the palette of the reef. One reason this partnership is so significant for reef ecology is that it makes reef building possible in nutrient-poor tropical waters. The tropical ocean is often described as a marine desert. Its warm, clear, sunlit waters contain very few dissolved nutrients. Without the photosynthetic input from zooxanthellae, a coral polyp would have to rely entirely on catching zooplankton with its tentacles, a process that provides enough energy for survival, but not nearly enough for the rapid calcification needed to build and maintain a reef in an environment where erosion is constantly at work. It turns out that the zooxanthellae are not a single species. The family Symbiodiniaceae contains many genetically distinct types, different clades and strains that vary in their photosynthetic efficiency, their heat tolerance, and their chemical relationship with their coral hosts. Scientists have identified at least 9 major clades, labeled A through I, with hundreds of distinct types within them. This diversity matters enormously for reef resilience. Different types of zooxanthellae have different tolerances for elevated water temperatures. Clade D zooxanthellae, for example, are generally more heat tolerant than clade C. They can continue photosynthesizing at temperatures that would cause clade C to break down and be expelled from coral tissue. Corals harboring clade D may therefore be better able to survive warm water events that cause bleaching in corals with less heat-tolerant zooxanthellae. One reason scientists are actively researching the zooxanthellae is precisely this variation in thermal tolerance. The possibility of transferring more heat-tolerant algal strains into coral species that are currently vulnerable to bleaching. essentially changing the coral's internal partner for one better suited to warmer conditions, is one of the more actively studied approaches to reef restoration. The evolutionary origins of this partnership are still being worked out. It is thought that the coral zooxanthellae symbiosis first evolved approximately 240 million years ago, around the time when modern reef-building corals appeared. Though the exact sequence of events that led to a dinoflagellate alga taking up permanent residence inside an animal's cells remains a subject of active research, what is not in question is the significance of the partnership. Without zooxanthellae, reef-building corals as we know them would not exist. The reefs would not exist. The 25% of marine species that depend on reef habitats would not have the ecosystem they depend on. The white beaches, the coastal protection, the fisheries, the medicines derived from reef organisms, all of it traces back to this intimate cellular partnership between a coral animal and a single-celled alga. In a sense, the coral reef, the largest biological structure on Earth, is the product of a relationship between two organisms that are individually invisible. Interestingly, some coral species can survive without their zooxanthellae for limited periods by feeding exclusively on zooplankton. But this is a temporary and energetically costly strategy. Without the photosynthetic input from their algal partners, corals cannot maintain the growth rates needed to keep pace with the biological and physical erosion that continuously works against the reef structure. The energy provided by zooxanthellae does not only sustain individual coral colonies. Over thousands of years, that energy has accumulated as calcium carbonate, layer upon layer, generation upon generation, into the reef structure itself. The reef is, in a very literal sense, the physical accumulation of millions of years of this partnership. Every meter of reef limestone is the product of zooxanthellae harvesting sunlight and passing its energy to polyps who converted it into stone. That stone, that accumulated geological legacy of a biological partnership, is what we will look at next. How reefs are built over deep time, how large they become, and what they contain within their ancient structure. That is where we are going next. A coral reef looks, from the outside, like a structure of modest depth. If you swim along the face of a reef wall, the vertical seaward edge of a reef dropping away into deep water, what you see is perhaps 10 or 20 meters of living coral surface. A beautiful, complex, inhabited wall of calcium carbonate covered in coral colonies of many species, with fish moving through and around every projection and crevice. It is impressive, but it gives no real sense of what lies behind it. Behind the living surface of a reef is a thickness of dead coral skeleton that can reach hundreds of meters, in some places, thousands of meters. The living reef is only the outermost skin of a structure built by uncountable generations of coral polyps over timescales that dwarf anything in recorded human history. The process of reef building is straightforward in its basic mechanics, even if extraordinary in its cumulative outcome. A coral polyp settles on a suitable hard surface, a rock, a piece of dead coral skeleton, and begins secreting calcium carbonate. Over its lifetime, it adds a few millimeters or a few centimeters to the structure. It reproduces, spawning daughter polyps that settle nearby. It eventually dies, and its skeleton becomes the foundation for the next generation. This cycle repeats across countless individuals, across thousands of years, and the reef grows. Different coral species grow at different rates. Branching corals, the staghorn and table corals of the genus Acropora, can add up to 20 centimeters per year under ideal conditions. These fast growers build the complex three-dimensional frameworks of shallow reef zones where light is abundant and competition for space is intense. Massive dome-shaped corals, the porites and diploria species, grow only 1 to 2 centimeters per year, but they can live for centuries. Their slow, patient growth-building columns and domes that resist wave energy and provide the structural backbone of the reef. Over thousands of years, these two strategies, fast branching growth and slow, massive growth, combine to produce the complex layered architecture of a mature reef system. Coral reefs take three main forms, each representing a different relationship between reef growth and the geological substrate beneath it. A fringing reef grows directly from the shoreline of an island or continent. The reef is attached to the land, its outer edge facing the open ocean. A barrier reef is separated from land by a lagoon of open water. The original fringing reef has continued to grow outward as the land has slowly subsided, leaving a gap between the reef and the shore. And an atoll is a ring of reef surrounding a lagoon with no visible land at its center. The explanation for how atolls form is one of the more elegant ideas in the history of natural science, and it was proposed in 1842 by Charles Darwin, based on observations made during the voyage of the Beagle, without his ever having been able to dive or drill into a reef. Darwin proposed that atolls are the final stage in the life cycle of a volcanic island. A new volcanic island rises from the ocean floor, and a fringing reef begins to grow around its shores. Over millions of years, the volcanic island slowly subsides, cooling and contracting as the magma beneath it solidifies. As the island sinks, the reef continues to grow upward, keeping pace with the rising sea level relative to the sinking land. Eventually, the island sinks below the surface entirely, leaving only the reef, now a ring of coral surrounding the lagoon that once covered the island's summit. Darwin's theory predicted that if you drilled through an atoll, you would find coral limestone extending all the way down to a volcanic base. The technology to test this prediction did not exist in his lifetime. Nearly a century later, in 1952, drilling at Eniwetok Atoll in the Marshall Islands found coral limestone extending 1,400 meters below the surface, precisely as Darwin had predicted, before hitting the volcanic rock beneath. The theory that Darwin had proposed from first principles and logical inference alone was confirmed. The Great Barrier Reef of Australia is the largest coral reef system on Earth, stretching approximately 2,300 kilometers along the northeast coast of Australia, comprising approximately 2,900 individual reefs and 900 islands, and covering an area roughly the size of Italy. It is the largest structure on Earth built by living organisms. It is visible from space, and it has been growing in its current location for approximately 500,000 years. Though the individual corals that make up its surface are much younger, the reef system itself is ancient beyond ordinary comprehension. Not all coral reefs grow in warm, sunlit tropical waters. Cold water coral reefs exist at depths of 200 to 6,000 meters, in water where no sunlight penetrates and temperatures are close to freezing. These reefs are built by corals that harbor no zooxanthellae. Without photosynthetic partners, cold water corals capture all their energy by filtering organic particles and zooplankton from the water currents that flow through and around the reef. The Røst Reef off the coast of Norway, one of the world's largest known cold water coral systems, extends approximately 35 kilometers at depths of 300 to 400 meters in water that never sees light. Cementing all reef structures together, both tropical and cold water, is a group of organisms often overlooked in descriptions of reef building. Coralline algae are calcareous red algae that grow encrusting surfaces, secreting calcium carbonate as they grow and binding the individual structural elements of the reef into a coherent whole. Without coralline algae, the physical integrity of reef structure would be significantly reduced. The reef crest, the shallowest, most wave-battered part of a reef, is dominated by coralline algae precisely because of its extraordinary resistance to mechanical stress. Scientists have found that the calcium carbonate skeletons of massive corals contain growth rings, annual bands of slightly different density, visible in cross-section, each one representing a year of the colony's life. These growth rings record, in their chemical composition, information about the conditions of the water in which they formed, its temperature, its salinity, its acidity, its productivity. A massive coral is a library, centuries of ocean history encoded in its skeleton, readable by researchers with the right instruments. Within that ancient accumulated structure, there is more life than anything visible on the surface suggests. And the first hint of that hidden life comes when the sun goes down and the reef begins its nightly transformation. That is where we are going next, at some point in the late afternoon, as the angle of sunlight changes and the water above the reef shifts from bright blue to something deeper and more golden. A transition begins. It happens gradually. The fish that have been active through the daylight hours, the butterfly fish picking at coral surfaces, the parrotfish biting chunks of skeleton, the surgeonfish grazing algae in steady passes across the reef, begin to slow their feeding and drift toward shelter. Crevices and overhangs that were empty during the day now fill with resting fish. A large grouper that spent the morning hunting in open water settles beneath a coral head and becomes still. A school of antheas that hovered in the water column above the reef descends and disappears into the reef structure. And as the day shift settles in for the night shift emerges. Cardinal fish appear from the caves where they sheltered during the day, their large eyes adapted for low-light hunting. Squirrelfish and soldierfish emerge from crevices, moving with the careful, deliberate motion of animals that have learned to navigate in near darkness. Octopuses flow out of rocky dens, changing color as they move from surface to surface. Hunters of extraordinary sensory sophistication, active in a world where vision alone is insufficient. And then, if you were to look very closely at the coral surface itself, at the surface that appeared textured and still during the day, you would notice something remarkable. It is moving, not dramatically, not in a way that announces itself, but across every square centimeter of coral surface, from the shallowest reef flat to the deepest colonies reachable by light. Tiny structures are emerging from the calcium carbonate cups in which they spent the day. Coral polyp tentacles, pale, translucent, each one bearing its cargo of stinging nematocysts, are extending into the water. The coral is feeding. During the day, the zooxanthellae within coral tissue photosynthesize, providing most of the coral's energy from sunlight. But zooxanthellae cannot provide everything. Coral polyps also need nitrogen, phosphorus, and other nutrients that photosynthesis alone does not supply. They obtain these by catching zooplankton, tiny animals that drift through the water with their tentacles. And zooplankton are most available at night when they migrate upward from deeper water toward the reef in a daily vertical movement driven by the cycles of light and dark. Scientists have found that a healthy reef at night is blanketed in this feeding activity. Millions of polyps extended across the reef surface, each one a tiny predator reaching into the current. Seen under underwater torchlight, a coral colony in full nocturnal extension looks covered in small pale flowers, an entirely different visual impression from the same colony by day, when the polyps are retracted and the surface appears as decorated stone. The soundscape of the night reef is one of its most distinctive features. A healthy coral reef produces a constant crackling sound, a continuous, textured noise that has been described as resembling the sound of frying oil or the crackling of a wood fire from a distance. This sound is produced primarily by snapping shrimp, small crustaceans that close a specialized enlarged claw so rapidly that a cavitation bubble forms in the water ahead of the claw, then collapses with a sharp pop. Billions of snapping shrimp, inhabiting every crack and crevice of the reef, produce this crackling continuously, and the collective sound is audible several kilometers away to a listener equipped with an underwater microphone. This sound is more than ambient noise. Researchers have found that fish larvae drifting in the open ocean use the sound of reef crackling as a navigational cue, orienting toward the sound to locate reef habitat from distances of several kilometers. The reef announces its presence acoustically in the dark ocean, and the larvae that will become its next generation of fish residents navigate toward that sound. In experiments where recordings of healthy reef sounds have been played through underwater speakers near degraded reef areas, the number of larval fish and invertebrates settling in those areas has increased measurably. The night reef also glows. Bioluminescence, the production of light by living organisms through chemical reactions, is common among reef organisms, particularly those inhabiting the darker zones within and beneath the reef structure. Some species produce steady blue or green light. Others flash in response to disturbance. The combined effect in a reef cave at night, seen by a diver with no artificial light, is a subtle luminosity, a faint inner light of biological origin that is one of the more quietly remarkable experiences available in the natural world. The shift from day to night on a reef is not simply a change in lighting. It is a wholesale change in community. The species present in the water column and on the reef surface at midnight are almost entirely different from those present at noon. The feeding relationships, the predator-prey dynamics, the social behaviors, all of them shift with the light. And yet, the reef remains equally busy, equally productive. The 24-hour rhythm of the reef is a complete ecological cycle, with the night shift doing work as essential to the reef's function as anything done by day. One reason the nighttime reef is particularly significant for reef recovery and reproduction is that it is during the night that the most spectacular biological event associated with coral reefs occurs. Not every night, only on certain nights, at a specific time of year, under conditions that must align with extraordinary precision. That event is the mass coral spawning, an annual renewal of the reef that has been happening on the same reefs for hundreds of thousands of years. That is where we are going next. Once a year, on 1 or 2 nights, a coral reef does something that has been described as one of the most synchronized biological events on Earth. It reproduces, all of it at once, in a single night. The mass spawning of coral reefs is not a gradual process spread across weeks or months. It is concentrated into hours, sometimes less, on specific nights that the reef has been building toward all year. And when it happens, the water above the reef transforms into something that resembles, more than anything else, a slow-motion blizzard falling upward. The triggers for mass spawning are multiple and must align precisely. Water temperature must have warmed to a threshold that signals the end of summer in the southern hemisphere or spring in the north. The lunar cycle must be at the right phase, typically a specific number of days after the full moon, when tidal conditions favor the dispersal of eggs and sperm into the open ocean rather than back toward the reef. Light levels in the hours before spawning must follow the gradual decline of a clear evening rather than being interrupted by cloud cover. When all these conditions are met simultaneously, something in the biochemistry of the corals signals readiness, and the spawning begins. On the Great Barrier Reef, mass spawning typically occurs on 1 or 2 nights in October or November. In the southern spring, a few days after the full moon. The precise timing has been documented since 1981, when researchers Carden Wallace and colleagues first described the phenomenon, a discovery that was at the time genuinely surprising, since it had previously been assumed that coral reproduction was a continuous and largely unsynchronized process. The spawn itself begins after dark. Individual coral polyps throughout the reef begin releasing bundles of reproductive material, tiny packets that contain both eggs and sperm, wrapped together in a buoyant package of lipids that causes them to rise slowly through the water column. These bundles are released in extraordinary numbers from every coral colony across the reef simultaneously, from the shallowest reef, flat, to the deeper slopes, from one end of a reef system to the other. As the bundles rise, the water above the reef fills with them. Millions, then hundreds of millions, then billions of tiny pale spheres rising toward the surface in a slow, continuous upwelling. Underwater, the visibility drops within minutes as the density of spawning material increases. The water takes on a milky, clouded quality, the pale bundles rising in every direction, catching the light of a diver's torch like an inverted snowstorm in very slow motion. Above the surface, the spawning slick can sometimes be visible as a pale film on the water, detectable from a boat, and in drone footage of the event, visible as a brightening of the sea surface across hundreds of kilometers. At the surface, the bundles break apart. Eggs and sperm from different coral colonies mix in the water. The simultaneous spawning of all colonies in a reef system, ensuring that cross-fertilization between genetically distinct individuals is possible. The mixing is critical. A coral colony spawning alone would fertilize its eggs only with its own sperm, producing genetically identical offspring. The mass spawning ensures genetic mixing across the entire reef population. Fertilized eggs develop into free-swimming larvae called planulae, tiny oval cells covered in cilia, barely visible to the naked eye, drifting in the plankton for days to weeks. During this planktonic phase, the larvae are carried by ocean currents, sometimes traveling considerable distances from the reef where they were spawned. Eventually, a planular larva settles on a suitable hard substrate and undergoes metamorphosis, transforming from a free-swimming larva into the first polyp of a new coral colony. What cues a planula to settle in one location rather than another is not completely understood. Researchers believe that chemical signals from coralline algae, which cover suitable settlement surfaces, play a role, as do the microbial communities on different substrates and possibly acoustic cues from the reef itself. The selection of a settlement site is one of the most consequential decisions in the life of a coral. The entire future colony will grow from this point. And yet, the mechanism by which tiny larvae make that decision remains an active area of research. The mass spawning attracts extraordinary visitors to the reef. Whale sharks, the largest fish on Earth, reaching 12 meters or more in length, visit the Great Barrier Reef during spawning events, specifically to feed on the vast quantities of coral eggs near the surface. These gentle filter feeders move slowly through the spawning slick, their enormous mouths open, straining the concentrated nutrients from the water. The whale shark visits are now sufficiently predictable that they have become the focus of scientific monitoring programs, and the timing of whale shark aggregations has become an additional indicator of spawning timing. The significance of the mass spawning extends beyond individual reproduction. It is the primary mechanism by which damaged or depleted areas of reef receive new coral colonies. Larvae from a healthy part of the reef drifting to a degraded area and settling there, beginning the slow process of rebuilding. The health and success of the annual spawning is therefore directly connected to the long-term resilience of the reef as a system. There is something worth sitting with for a moment in the image of the mass spawning. Billions of organisms, each one independently tiny and individually insignificant, coordinating a reproductive event of such precise timing and such enormous scale. An event triggered by the alignment of temperature and moonlight and day length into a specific combination that occurs on only 1 or 2 nights per year. The reef that produces this event has been doing so for hundreds of thousands of years. The same reef system, the same annual spawning, the same snowstorm of eggs rising through dark water, before any human was there to witness it, before the Great Barrier Reef had its name, before the continent of Australia had been mapped by anyone outside it. The spawning continues, as it always has, as it will, we hope, for a very long time to come. But the spawning is only one of the hidden processes of the reef. Within the reef itself, inside its structure, not on its surface, there is another world that most visitors to a reef never see at all. That is where we are going next. Most people who visit a coral reef, who snorkel above it or dive along its face, experience the reef as a surface. They see what is visible on the outside: the coral colonies, the fish swimming among them, the sea fans and sponges and feather stars decorating the structure. It is beautiful and rich and alive, and it is easy to understand why coral reefs inspire such wonder in those who encounter them. But the surface of a reef is only the beginning. Within the reef, within the accumulated calcium carbonate structure built by generations of coral polyps over centuries and millennia, there is another world. A world of tunnels and caves and crevices and passages, of spaces carved by biological erosion and left by the irregular growth patterns of successive coral colonies, of chambers and corridors that extend deep into the reef matrix in every direction. Most of this interior is completely invisible from outside, and within it lives a community of organisms whose diversity may rival or exceed that of the visible surface. Scientists estimate that for every species visible on the surface of a coral reef, there may be 10 or more species living within the reef framework itself. The cryptofauna, organisms inhabiting the interior of the reef structure, are among the most poorly known components of the reef ecosystem, not because they are rare, but because they are so difficult to study. They live in spaces that are too small for divers to enter, too dark for standard photography to illuminate, and too structurally complex for systematic sampling. What researchers have found when they have extracted and carefully examined sections of reef framework has been startling. A single cubic meter of reef rubble, a small piece of the larger structure, can contain dozens of species of polychaete worms, crustaceans of many groups, mollusks, echinoderms, sponges, and organisms from nearly every major invertebrate phylum. The diversity within the reef is not less than the diversity on its surface. In many assessments, it is greater. The caves and overhangs within and beneath reef structures harbor specialized communities that are entirely different from the surface reef communities. Where the surface reef is dominated by zooxanthellid corals that require sunlight, the cave communities are composed of organisms that require no light at all. Cup corals, small solitary corals that harbor no zooxanthellae, cover cave walls and ceilings in dense aggregations, their tiny tentacles extended into the water flowing through. Sea fans spread their branching structures perpendicular to the current to maximize food capture. Sponges of many species, including some not found anywhere else on the reef, encrust every available surface in vivid colors ranging from bright orange to deep purple. These cave communities are sustained not by sunlight, but by water. The reef is not a solid structure. It is porous, threaded with passages through which water flows continuously, carrying dissolved organic matter, bacteria, and plankton that feed the filter-feeding organisms within. The cave inhabitants are, in effect, living off the productivity of the sunlit reef above and outside them, receiving a share of its bounty in the form of organic particles carried inward by the current. Bioluminescence is particularly common among the organisms that inhabit the darker zones of the reef interior. Many species produce their own light through chemical reactions. blue or green, bioluminescent light used for communication, for luring prey, or simply as a byproduct of metabolic processes. In a reef cave with no artificial illumination, the faint glow of bioluminescent organisms can be visible to a dark, adapted eye. A subtle, shifting luminosity produced by animals that have never seen sunlight. Below the shallow reef zone, familiar to most divers, the reef continues into a depth range that has only relatively recently been recognized as a distinct and important habitat zone. The mesophotic zone, from the Greek word for middle light, encompasses reef habitat between approximately 30 and 150 meters depth. Here, sunlight is present, but reduced, filtered by the water column to a fraction of its surface intensity. The corals and other organisms adapted to this zone are distinct from those of the shallower reef, often more plate-like in form, maximizing their surface area to capture the limited available light. Researchers have found that the mesophotic zone harbors species not seen in shallower reef surveys. Organisms that are either specialists of this depth range or populations of shallow reef species that extend further into the mesophotic than expected. Some researchers believe that mesophotic reefs may serve as refugia for certain species when shallow reefs are affected by bleaching events or other stressors. Deeper water being slightly cooler and potentially buffering organisms from the elevated temperatures that cause bleaching above. Whether the mesophotic zone can truly serve as a source of recovery for damaged shallow reefs, whether larvae produced at depth can successfully colonize shallow areas and rebuild damaged populations, is an active question in reef science. The answer may vary by species and by location, and understanding it better is considered important for reef conservation planning. The picture that emerges from all of this is of a reef as a three-dimensional structure of extraordinary depth and complexity. Not a flat backdrop for a fish community, not a decorated rock surface, but a multi-layered world extending vertically through the water column and horizontally within the reef matrix, inhabited at every level by organisms that are doing the work of the reef ecosystem in their particular way. The reef you can see from the surface is, in a sense, the advertisement for something much larger. The interior of the reef, the hidden architecture, the cave communities, the cryptofauna, the mesophotic depths, is the substance. And inhabiting this three-dimensional world is a community of organisms whose social lives are, in their own way, as complex and intricate as anything visible in the more familiar social animals of the land. That social complexity is where we are going next. A coral reef is not simply a feeding ground. It is a society. Not in the metaphorical sense in which we might describe any complex ecosystem as society-like, but in a more literal sense. A community of organisms with established roles, fixed locations, recognized relationships, and forms of communication that allow individuals to coordinate their behavior with other individuals in ways that benefit both parties. This social complexity is not concentrated in any one group of reef organisms. It is distributed across the reef, visible in the behavior of fish, of crustaceans, of cephalopods, and even of the coral polyps themselves. But it is perhaps most clearly expressed in one particular institution of the reef that has been studied since the 1950s, and that continues to reveal new dimensions of complexity. The cleaning station. A cleaning station is a fixed location on the reef, typically associated with a prominent coral head or a particular piece of reef structure, where specific small fish or shrimp provide a service to larger visiting fish. The service is parasite removal. External parasites, dead tissue, and food debris accumulate on the skin, gills, and mouths of reef fish, and the cleaning station providers, typically cleaner wrasse or cleaner shrimp, remove these materials by picking them off with their specialized mouths or claws. The cleaner wrasse, a small, distinctively colored fish with a bright blue and yellow body pattern, signals its role through both its coloration and a specific undulating dance that it performs at or near the cleaning station. Client fish recognize this signal and approach the station, adopting a characteristic posture, hovering motionless, often at an unusual angle, sometimes changing color slightly, that signals their willingness to be cleaned. What happens next is one of the more remarkable behavioral interactions on the reef. Large predatory fish, moray eels, groupers, sharks, and others that would, under any other circumstance, eat a small fish the size of a cleaner wrasse, allow themselves to be cleaned with complete passivity. The cleaner wrasse enters the mouth of a fish that could consume it in a single movement and picks parasites from between its teeth and from its gills, while the client fish remains still. The cleaning station is, in effect, a zone of truce, a place where the normal predator-prey relationships of the reef are suspended by mutual agreement. Reef fish, specifically the blue-streak cleaner wrasse, have been shown in research published in 2019 to recognize themselves in mirrors. When a mark is placed on the body of a cleaner wrasse in a position visible only in a mirror, the fish attempts to remove the mark by rubbing that part of its body, a response interpreted as self-recognition. This finding has generated significant scientific debate, but if confirmed, it would represent a level of self-awareness not previously attributed to fish. The communication system of reef fish extends well beyond what human observers can easily perceive. Many reef fish have patterns on their bodies that are visible only in ultraviolet light, patterns invisible to human eyes, but clearly visible to other reef fish, whose visual systems extend into the ultraviolet range. These UV patterns appear to play roles in species recognition, mate selection, and possibly territory signaling. The reef is, in this sense, overlaid with a layer of social communication that humans simply lack the sensory equipment to observe directly. Some of the most intricate partnerships on the reef involve not fish with fish, but fish with crustaceans. The goby shrimp partnership is among the best studied of these. A nearly blind pistol shrimp excavates and maintains a burrow in the sandy substrate at the base of reef structures, a burrow that provides shelter for both the shrimp and a small goby fish that lives at the entrance. The shrimp, unable to see well enough to detect approaching predators, maintains constant contact with the goby through its antenna resting against the goby's tail. When the goby detects danger, it flicks its tail, a signal the shrimp interprets immediately, retreating into the burrow while the goby darts in behind it. In calmer moments, the goby stands guard while the shrimp excavates, pushing sand out of the burrow entrance, working continuously to maintain the shared dwelling. The pistol shrimp itself deserves a moment of attention for one of the more remarkable physical phenomena associated with reef life. When the pistol shrimp closes its specialized enlarged claw at high speed, the water ahead of the claw is accelerated so rapidly that a cavitation bubble forms, a small vacuum bubble in the water. The collapse of this bubble generates a pressure wave that stuns or kills small prey items, and the temperature inside the collapsing cavitation bubble, measured by sensitive instruments, briefly approaches that of the surface of the sun. A small crustacean on a coral reef produces, in the space of a fraction of a millisecond, temperatures comparable to the solar surface. Using this capacity, not as a party trick, but as a hunting strategy, refined over millions of years. The damselfish, small, territorial, often brilliantly colored, adds another dimension to reef social complexity through what can only be described as farming. Certain damselfish species maintain carefully tended patches of algae on the reef surface, weeding out algae species they do not prefer, defending their plots aggressively against any intruder regardless of size, and creating a managed agricultural territory in the midst of the broader reef community. A damselfish farmer will charge at fish many times its own size, at octopuses, and at human divers who venture too close to its plot, a display of territorial confidence that seems comically disproportionate, but is rooted in a genuine ecological strategy. The social reef, then, is not a backdrop for individual animals going about their individual lives. It is a community with roles and relationships and communication channels and partnerships, some mutualistic, some competitive, some neutral, that together constitute a social fabric as rich as anything found in the more familiar social animals of the terrestrial world. Within this social world, some roles are defined by hunting, by the predatory strategies of animals evolved to exploit the extraordinary density of life that the reef provides. Those predators are where we are going next. A coral reef is, among other things, an extraordinarily productive hunting ground. The density of life on a reef, the sheer number of individual organisms per cubic meter of water and per square meter of reef surface, provides predators with feeding opportunities unmatched in most marine environments. And the reef has responded over millions of years of evolution by producing predators of extraordinary variety, sophistication, and effectiveness. Some of the reef's most formidable predators are among its smallest. The blue-ringed octopus is found on Indo-Pacific reefs and is approximately the size of a golf ball to a tennis ball. A small, soft-bodied cephalopod that hunts crabs and small fish in the crevices of the reef. What makes the blue-ringed octopus notable is not its size, but its venom. It carries tetrodotoxin, a neurotoxin that blocks sodium channels in nerve cells, preventing nerve impulses from firing, in quantities sufficient to paralyze or kill a human adult within minutes. The blue-ringed octopus does not produce this toxin itself. It harbors bacteria in its salivary glands that produce it. The vivid blue rings that give the animal its name become more intensely colored when the animal is threatened, a warning signal to potential predators that the visual display is associated with chemical danger. The cone snail, another reef predator of modest appearance but formidable capability, hunts fish and worms using a harpoon-like radula tooth that it can extend in any direction from its shell, including backward. The venom delivered by this tooth contains a complex cocktail of peptides called conotoxins, some of which interfere with nerve function with extraordinary specificity. One of these peptides, a compound that blocks specific pain receptor channels in nerve cells, has been developed as a pharmaceutical painkiller called ziconotide, which is approximately 1,000 times more potent than morphine for certain types of chronic pain and is used clinically where other pain management approaches have failed. The mantis shrimp occupies a different category of predatory capability entirely. There are approximately 500 species of mantis shrimp on coral reefs worldwide, and they fall into two broad groups: those that use sharp-bladed appendages to spear soft prey, and those that use heavily calcified club-like appendages to smash hard-shelled prey. It is the smashers that produce the most physically remarkable hunting strike in the animal kingdom. A mantis shrimp club accelerates at approximately 10,000 times the acceleration of gravity and strikes with the force of a bullet. The impact is sufficient to shatter crab shells, to break apart snail shells, and to crack aquarium glass, which is why mantis shrimp are sometimes called thumb splitters by people who keep marine aquariums. The mantis shrimp also sees the world in ways that remain incompletely understood. It has 16 types of photoreceptor in its eyes, compared to the 3 found in human eyes. This does not necessarily mean the mantis shrimp perceives 16 times more colors than humans. The processing appears to work differently, possibly as a rapid color identification system, rather than a nuanced color discrimination system. But the visual complexity of its eyes is without parallel in the animal kingdom, and what it actually experiences when it looks at a coral reef is, genuinely, something we cannot know. At the level of the larger reef ecosystem, the most ecologically significant predators are the reef sharks. Reef sharks, white tip, black tip, grey reef, and others depending on the ocean, are keystone predators. This means that their presence or absence has effects on the reef ecosystem that extend far beyond their direct predation. A keystone predator maintains balance in a food web by preventing any single prey species from becoming so abundant that it dominates the ecosystem at the expense of others. Remove the keystone and the food web reorganizes in ways that cascade through every level. Research comparing reefs inside and outside marine protected areas has found that reefs with intact shark populations show measurably higher coral cover than reefs where sharks have been removed by fishing. The mechanism runs through multiple steps. Sharks control populations of mid-level predators. Mid-level predators control populations of herbivorous fish. Herbivorous fish control algae growth on the reef. When sharks are removed, mid-level predators increase, herbivorous fish decrease, algae expands, and coral loses the competition for space on reef surfaces. Among the reef's predators, the hawksbill sea turtle occupies a particularly specific ecological role. Hawksbill turtles feed primarily on sponges, a food source that almost nothing else on the reef will eat, because sponges contain chemical defenses that deter most predators. Sponges can grow rapidly and, without hawksbill predation, have the potential to overgrow corals on reef surfaces. The hawksbill's consumption of sponges keeps sponge populations in check, maintaining space for coral growth. The lionfish, native to the Indo-Pacific but introduced to the Atlantic and Caribbean, almost certainly through the release of aquarium fish, illustrates what happens when a capable predator is placed in an environment where it has no natural checks. In the Atlantic and Caribbean, lionfish have no natural predators that regularly hunt them. Their populations have expanded rapidly, and they have significantly reduced populations of small reef fish in many areas, with potential cascading effects on reef herbivory and coral cover. The predators of the reef, from the tetrodotoxin-bearing blue-ringed octopus to the ecosystem-structuring reef shark, are not simply consumers. They are architects of the reef community, shaping its composition and dynamics through their presence and their hunting. But the reef's ecological balance depends as much on those that eat plants as on those that eat animals, and the organisms that graze the reef, that keep algae from overgrowing the coral, are as essential to the reef's health as any predator. That is where we are going next. There is a white sandy beach somewhere in the tropical world, perhaps one you have visited or one you have seen in a photograph, that is composed in significant part of fish excrement. Not entirely, not solely, but a substantial fraction of the fine white sand on many tropical beaches near coral reefs is calcium carbonate that was once part of a living coral skeleton, was bitten off by a parrotfish, passed through its digestive system, and was excreted as white sand. The parrotfish is not primarily eating coral. It is eating the algae that grows on and within the coral skeleton. The calcium carbonate of the skeleton is incidental. It is ingested along with the algae the parrotfish actually wants and excreted after the algae have been digested. A single large parrotfish can produce up to 90 kilograms of sand per year. Across a reef system with thousands of parrotfish, across thousands of years, this is a substantial geological contribution. The sandy floors of reef lagoons, the sandy beaches of reef islands, the fine sediment carried by ocean currents away from reefs. Much of this material has passed through parrotfish. The parrotfish is also doing something ecologically essential beyond sand production. By grazing the algae from coral surfaces and from within coral skeleton, it maintains clear, clean surfaces on which coral larvae can settle and new colonies can establish. A reef without parrotfish is a reef where algae accumulates unchecked on available surfaces, surfaces that might otherwise be colonized by new coral. Surgeonfish, known in the aquarium trade as tang, perform a similar ecological function. These laterally flattened fish graze constantly on algae, moving in schools across reef surfaces and keeping algae growth cropped to minimal levels. They have a specialized defensive weapon, a sharp spine at the base of the tail as hard as a scalpel that gives them their common name. But their primary ecological contribution is not defense, but feeding. The continuous, methodical, reef-wide removal of algae growth that would otherwise shade and smother the corals beneath. Sea urchins, particularly the long-spined sea urchin Diadema antillarum in Caribbean reefs, perform similar grazing on reef surfaces, particularly in areas where fish grazing is reduced. Diadema are nocturnal grazers, emerging from crevices at night to scrape algae from carbonate surfaces with their rasping mouthparts, maintaining the clean surfaces that coral requires for recruitment. In 1983, a pathogen, waterborne, spreading rapidly, still not definitively identified at the time, swept through the Caribbean. Within approximately 13 months, it had killed more than 93% of Diadema antillarum across the entire Caribbean Sea, one of the largest and most rapid mass mortality events ever documented for a marine organism. The ecological consequences were severe and lasting. Without the grazing pressure of Diadema, algae grew unchecked on Caribbean reef surfaces. Corals that had already been stressed by warming events and disease lost the competition for space to algae. Many Caribbean reefs shifted from coral-dominated states, in which coral covered the majority of available hard surface, to algae-dominated states, in which fleshy algae covered more surface than coral. This transition represented a fundamental change in the character of Caribbean reef ecosystems. 40 years later, Diadema populations in most Caribbean locations have not recovered to their pre-1983 levels. The shift in reef state that followed the die-off has persisted in many areas, demonstrating how a single ecological event, the loss of one species of grazer, can produce lasting changes to an entire regional ecosystem. Crown-of-thorns starfish, large, multi-armed, spiny echinoderms that feed directly on coral tissue, are another organism whose grazing is essential in small numbers but catastrophic in large ones. At normal population densities, crown-of-thorns starfish are a natural part of reef ecology, consuming some coral tissue and creating space for other species. At outbreak densities, which can reach dozens of starfish per square meter, they consume coral tissue faster than the reef can regrow it, leaving large areas of bare white skeleton behind as they move across the reef. Outbreaks of crown-of-thorns starfish have been linked to nutrient pollution. Excess nutrients from agricultural runoff cause blooms of the phytoplankton that juvenile crown-of-thorns starfish feed on, allowing more juveniles to survive to adulthood than would normally be the case, producing the population explosions that overwhelm reef coral. The damselfish farmer adds complexity to this grazing story. Certain damselfish species actively maintain territories on the reef surface, patches of algae that they weed, defend, and manage with an intensity that resembles, in its outcomes, if not its motivation, small-scale agriculture. By excluding other grazers from their territories, damselfish maintain patches of specific algae species that they prefer. These territories can accumulate algae that reduces the available space for coral, particularly in areas where damselfish densities are high. The balance between grazing and growth is one of the fundamental dynamics of a healthy reef. When grazing is adequate, algae is kept at bay, and coral has the space and light it needs to grow. When grazing is reduced, whether by overfishing of parrotfish and surgeonfish, by disease-killing sea urchins, or by nutrient pollution fueling algae growth that outpaces grazing capacity, the balance tips and the reef changes character. The grazers are not charismatic in the way that sharks or dolphins are charismatic. They do not inspire the same immediate wonder as a sea turtle or a manta ray, but their ecological importance is as fundamental as any more dramatic organism on the reef. Equally fundamental, and even less visible than the grazers, are the organisms that perform the chemical and physical engineering that makes the reef possible in the first place. That is where we are going next. There are organisms on a coral reef that do more ecological work than almost anything else on the reef, filtering more water, cycling more nutrients, contributing more to the physical integrity of the reef structure. And yet, they attract far less attention than the fish and turtles and sharks that draw most human interest to reef ecosystems. Some of them are not even animals. Begin with the sponges. Sponges are among the most ancient multicellular animals on Earth. Their basic body plan has existed for hundreds of millions of years, and their ecological function on coral reefs is so fundamental that without them, the reef would be a profoundly different and less healthy place. A sponge is, at its core, a filtering machine, a porous body through which water is continuously pumped, passing through increasingly fine channels where bacteria, organic particles, and dissolved organic matter are extracted and absorbed. A single barrel sponge, one of the largest species found on Caribbean and Indo-Pacific reefs, can filter approximately 50,000 liters of water per day. 50,000 liters. The water in a large residential swimming pool might be 30,000 liters. The sponge filters more water than that, continuously, every day of its life, removing bacteria and organic matter that would otherwise accumulate in reef waters and reduce the water clarity that corals depend on for their zooxanthellae photosynthesis. Scientists have found that sponges on coral reefs may cycle more nitrogen than all other reef organisms combined. Nitrogen, an essential nutrient for biological growth, enters coral reef systems in limited quantities and must be recycled efficiently to sustain the extraordinary productivity of the reef. Sponges process dissolved organic nitrogen in ways that make it available to other reef organisms, and the scale of this processing is so large that sponges are now understood as a critical component of reef nutrient dynamics, rather than simply passive filter feeders. Coralline algae, calcareous red algae that grow encrusting surfaces throughout the reef, are equally important, though in a completely different way. Rather than filtering water, coralline algae cement the reef. The individual structural elements of a coral reef, the branches of dead acropora, the domes of dead porites, the rubble produced by storm damage and biological erosion, would, without some binding agent, be an unstable pile of calcium carbonate fragments rather than a coherent structural framework. Coralline algae grow over these surfaces, encrusting them and binding them together into a solid structure. The calcium carbonate that coralline algae secrete is the mortar between the bricks of coral skeleton, and without it, the reef would be far less structurally sound. Interestingly, the part of the reef most exposed to wave energy, the reef crest, the shallowest zone where ocean swells strike the reef with their full force, is dominated by coralline algae rather than by coral. The wave energy that would destroy most coral colonies is endured by the coralline algae, whose dense encrusting structure is among the most mechanically resistant biological materials in the ocean. Halimeda, a green alga with a jointed, calcified structure, contributes to the reef in yet another way. As Halimeda grows, it produces calcium carbonate nodules. When it dies, these nodules sink and accumulate as sediment in reef lagoons and on reef flats, contributing to the physical substrate of the reef environment and ultimately to the formation of lagoon floors and reef islands over geological time. At the microscopic level, cyanobacteria, the same group responsible for producing Earth's oxygen billions of years ago, perform an essential service on coral reefs. They fix atmospheric nitrogen, converting nitrogen gas from the water into ammonia and other compounds that can be absorbed and used by reef organisms. In the nutrient-poor waters of tropical reef environments, this nitrogen fixation is a critical input, supporting the productivity of an ecosystem that would otherwise be severely nitrogen limited. Within the tissue of the coral animals themselves lives another invisible community, the coral microbiome. The tissue of a single coral colony harbors a complex community of bacteria, viruses, fungi, and other microorganisms, each performing different biochemical functions in relationship to the coral host. Some produce compounds that protect the coral from pathogens. Some contribute to the breakdown of organic matter within coral tissue. Some play roles in calcification, the process by which the coral deposits its calcium carbonate skeleton. Through the chemical gradients, they help maintain around the growing skeleton. Researchers have found that the coral microbiome changes under stress. Corals that are bleaching or diseased have different microbial communities from healthy corals, and this has opened the possibility of using the microbiome as both a diagnostic tool for reef health and a potential target for intervention. Introducing beneficial microorganisms to stressed coral has been proposed and tested as a potential way to improve coral resilience, though this research is still in its early stages. The reef, then, is not simply a community of visible organisms occupying a calcium carbonate structure. It is a chemical and biological system maintained by organisms operating at scales from the molecular to the meter, performing functions from nitrogen cycling to structural cementing to water filtering that together sustain the extraordinary productivity of the ecosystem above them. The reef is a factory as much as a garden, and the factory is embedded in a larger production system that extends beyond the reef's physical boundaries into the coastal landscape around it. That broader connected world is where we are going next. A coral reef does not exist alone. This is easy to overlook when thinking about reefs because the reef itself is so visually and biologically dominant that it can seem self-contained, a world complete in itself, needing nothing outside its own boundaries. But the reef is embedded in a coastal ecosystem that includes two other habitat types, each as productive and ecologically significant as the reef itself, and each connected to the reef in ways that mean the health of one affects the health of all three. Those two habitats are mangrove forests and seagrass beds. Mangrove forests are dense tangles of salt-tolerant trees growing in the shallow, muddy coastal waters of tropical and subtropical regions. Their roots, arching prop roots in many species, or upward projecting pneumatophores, create a three-dimensional underwater structure of extraordinary complexity. The spaces within and beneath the mangrove root system are sheltered, productive, and rich in invertebrate life. They are, in the ecological sense, nurseries. Many of the fish that live as adults on coral reefs spend the early phases of their lives in mangroves. Juvenile snappers, groupers, grunts, and many other reef fish species use mangrove root systems as shelter from the predators. They are not yet large enough to escape in open water. The mangroves provide food. Invertebrates are abundant in the organic rich mangrove sediment. And safety. The structural complexity of the root system makes pursuit by larger predators difficult. After months or years in the mangroves, juvenile fish move out to seagrass beds and eventually to the reef as they grow and mature. Seagrass beds, meadows of aquatic flowering plants growing in shallow coastal waters, serve as feeding grounds for both juvenile and adult reef organisms. Many reef fish spend significant feeding time in adjacent seagrass beds, consuming the invertebrates that live within and on the seagrass. Seagrass beds also serve as habitat for organisms that are themselves ecologically important to the broader reef system. Green sea turtles are among the primary grazers of seagrass. Their selective feeding, choosing specific seagrass species and grazing particular areas repeatedly, has been found to maintain the diversity and productivity of seagrass beds in ways analogous to the way that grazers maintain grasslands on land. Without sea turtle grazing, seagrass beds can become dominated by a few species and accumulate dead plant material that reduces overall productivity. Dugongs, large, slow-moving marine mammals related to manatees, are another important seagrass grazer. They feed by uprooting seagrass plants, leaving characteristic feeding trails across the seagrass bed that are visible from above. Dugongs are found in tropical coastal waters adjacent to reef systems across the Indo-Pacific, and their grazing behavior appears to maintain seagrass diversity in ways that benefit the broader coastal ecosystem. The connectivity between mangroves, seagrass, and reef means that damage to any one of these habitats affects the others. When mangrove forests are cleared for coastal development, a process that has eliminated large areas of mangrove habitat across the tropics, the nursery function they provide is lost. Fewer juvenile fish enter the seagrass and reef systems. Adult fish populations on adjacent reefs may decline in response to reduced recruitment from the mangroves. The value of the reef ecosystem extends well beyond its ecology. Coral reefs protect approximately 150,000 kilometers of tropical coastline from wave energy and storm surges. The physical structure of the reef dissipating ocean wave energy before it reaches the shore, reducing erosion and flooding risk for coastal communities. This protective function has economic value that has been estimated in the hundreds of billions of dollars annually. Approximately 500 million people worldwide depend on reef fish as a primary protein source, making coral reef fisheries one of the most important food security resources in the world, particularly for island and coastal communities in the tropics with few alternative protein sources. The medicinal value of reef organisms is less widely known, but equally significant. The compound that led to the development of AZT, one of the early treatments for HIV, was derived from a chemical found in a Caribbean reef sponge in the 1950s. Ziconotide, a painkiller derived from cone snail venom, was approved for clinical use in 2004 and provides relief for patients with chronic pain for whom other treatments have failed. Researchers are continuing to investigate reef organisms as potential sources of pharmaceutical compounds, reasoning that organisms that have evolved powerful chemical strategies for defense and predation over millions of years may have developed biochemical solutions to problems that human medicine has not yet addressed. The acoustic health of a reef, the soundscape it produces, is now being used as a monitoring tool. The crackling, clicking, and humming of a healthy reef is measurably different from the quiet of a degraded one. Researchers deploying underwater microphones on reefs can assess reef health by listening to the soundscape, and the playing of healthy reef sounds near degraded areas has shown promise as a technique for attracting larvae and accelerating reef recovery. The reef and the broader coastal ecosystem it is part of sustain extraordinary natural and human value. Understanding this value, not just the ecology of the reef itself, but the broader system of which it is a part, is essential to understanding what a reef actually is. And the history of how humans have come to that understanding is a story of patient observation, elegant theory, and continuous discovery. That history is where we are going next. For most of human history, corals were not understood to be animals. This is understandable. A coral colony does not move. It does not appear to feed in any visible way. It has the color and texture of a plant or a mineral. It grows in the way that crystals grow, slowly, without apparent intention or behavior. And for centuries, natural philosophers and scientists classified corals accordingly, as plants, or as some intermediate category between plant and mineral. The first serious challenge to this classification came in 1725, when a French physician named Jean-André Peysonnel observed corals closely enough to describe their polyp structure, noting that they produced eggs and had the characteristics of animals rather than plants. His paper was not well received. The scientific establishment of the time rejected his conclusions, and it was not until 1727 that René Antoine Virchow de Réaumur made a similar argument more successfully, beginning the gradual acceptance that corals are indeed animals, members of the phylum that would eventually be called Cnidaria. Understanding that corals are animals did not immediately clarify how they build their structures or what sustains their growth. The relationship between corals and the algae living within them, the zooxanthellae partnership that provides most of the coral's energy, was not fully understood until the 1970s and 1980s, despite the algae themselves having been described much earlier. The idea that a coral derives most of its energy not from catching food, but from the photosynthesis of internal algae was, for many years, not fully appreciated. The question of how atolls form was addressed by one of the 19th century's most productive scientific minds in one of the more elegant examples of scientific inference from indirect evidence in natural history. Charles Darwin, during the voyage of the Beagle from 1831 to 1836, observed coral reefs in the Pacific and Indian Oceans. He could not dive on them. The technology for extended underwater observation would not exist for another century. He could not drill into them, but from observations of reef structure, from the patterns of fringing reefs around volcanic islands, from the geometry of barrier reefs and atolls, he developed a theory of atoll formation that was published in 1842 and that proved to be entirely correct. Darwin proposed that atolls are the final stage in a sequence beginning with a fringing reef around a volcanic island. As the island subsides over geological time, the reef grows upward to keep pace with the rising water, eventually producing a barrier reef as a lagoon forms between the reef and the sinking land, and ultimately an atoll as the island disappears entirely below the surface. The theory required that coral limestone of atoll thickness extend all the way down to a volcanic base, a prediction that could not be tested until drilling technology permitted it. Nearly a century after Darwin published his theory, drilling at Eniwetok Atoll in 1952 found coral limestone extending 1,400 meters below the surface before hitting the volcanic rock beneath. Darwin had been right. The opening of coral reefs to extended human observation came with the development of the aqua lung by Jacques Cousteau and Émile Gagnon in 1943. For the first time, divers could remain underwater for extended periods, long enough to observe reef communities in their full context, to follow fish through their daily activities, to examine the reef surface in detail and document what lived on it. Cousteau's subsequent films and books, The Silent World, The Living Sea, and the long-running television series The Undersea World of Jacques Cousteau, brought the visual reality of coral reef ecosystems to global public audiences for the first time, transforming what had been a largely scientific subject into a subject of broad cultural fascination. The first scientific expedition dedicated specifically to coral reef study was the Great Barrier Reef Expedition of 1928 to 1929, led by C.M. Young and based at Low Isles on the northern Great Barrier Reef. This expedition established many of the basic ecological parameters of reef biology, but the scale and complexity of reef science had only begun to be appreciated. The 1980s brought a turning point in reef science that was, at the time, unexpected. The 1982 to 1983 El Niño event produced sea surface temperatures significantly above normal across large areas of the tropical Pacific and Indian Oceans, and for the first time, scientists documented widespread coral bleaching, the expulsion of zooxanthellae from corals under thermal stress, affecting reefs across multiple reef systems simultaneously. In 1998, a stronger El Niño produced the first truly global bleaching event, affecting reef systems in 3 ocean basins simultaneously and demonstrating that the phenomenon could operate at a planetary scale. Remote sensing from satellites, using instruments that detect sea surface temperature and water optical properties, became an important tool for reef monitoring from the 1990s onward, allowing scientists to track thermal stress conditions at global scales and identify areas at risk of bleaching before field teams could be deployed. In the early 2000s, the Coral Triangle, encompassing the waters of Indonesia, the Philippines, Malaysia, Papua New Guinea, the Solomon Islands, and Timor-Leste, was formally identified and named as the Global Center of Marine Biodiversity. The surveys that identified it found approximately 600 species of reef-building coral within its boundaries, compared to approximately 70 in the Caribbean. A difference that reflects both the age of the coral triangle as a center of marine evolution and its position at the convergence of multiple ocean current systems. The history of reef discovery is ongoing. New reef systems continue to be found, particularly cold water reef systems at depth, whose global extent is still not fully known. New species are described from reef surveys every year. New dimensions of reef ecology, the social complexity of reef fish, the role of the microbiome, the acoustic monitoring of reef health, continue to be revealed by research that was not possible a generation ago. The story of how humans came to know the reef is a story of patient observation across centuries, of elegant theory confirmed long after it was proposed, of technology enabling access to a world that had existed unobserved throughout all of recorded human history. It is a story still being written, and the current chapter of that story includes both the challenges that reefs face and the science being done in response to those challenges. That is where we are going next. Coral reefs are facing challenges. This is worth saying clearly and calmly, without alarm, but without minimization, because understanding what is happening to reefs is part of understanding what reefs are and why they matter. The most widely discussed challenge is coral bleaching. Bleaching occurs when the water temperature around a coral reef rises above the threshold that the zooxanthellae within coral tissue can tolerate, typically about 1 degree Celsius above the average seasonal maximum, sustained for several weeks. Under this thermal stress, the zooxanthellae become dysfunctional. They begin producing compounds that are toxic to the coral cell rather than the sugars that sustain it. In response, the coral expels its zooxanthellae, pushing them out of its tissue. Without the zooxanthellae, the coral tissue becomes nearly transparent. The white calcium carbonate skeleton beneath shows through the thin, colorless tissue, and the coral appears white, bleached. This is not the coral skeleton exposed by death. The coral is still alive, its tissue is still present, but it has lost the partnership that provides up to 90% of its energy, and it is now entirely dependent on catching zooplankton with its tentacles, a far less efficient energy source. A bleached coral can survive. If water temperatures return to normal relatively quickly, within weeks rather than months, the zooxanthellae can recolonize the coral tissue and the coral can recover. Many corals do recover from bleaching events, but if elevated temperatures persist, the coral cannot maintain its tissue without adequate energy. It starves. The tissue dies and sloughs off, exposing the white skeleton beneath. But this time, the white is not bleaching. It is death. Algae begins to colonize the bare skeleton within days, and what was a living coral colony becomes an algae-covered rock. The first documented mass bleaching events occurred during the El Niño of 1982 to 1983. The first truly global bleaching event, affecting reef systems in the Pacific, Indian, and Atlantic Oceans simultaneously, occurred in 1998 and killed an estimated 16% of the world's coral reefs. Subsequent bleaching events have affected various reef systems in subsequent years, and the frequency and severity of thermal stress events affecting reefs has increased as ocean temperatures have risen. Ocean acidification adds a different and compounding challenge. The ocean absorbs a significant fraction of the excess carbon dioxide in the atmosphere, currently approximately a quarter of annual carbon dioxide emissions. And when carbon dioxide dissolves in seawater, it forms carbonic acid, reducing the pH of the water. More acidic water contains fewer carbonate ions, the building blocks of calcium carbonate that corals use to build their skeletons. As ocean acidity increases, the process of calcification, the building of calcium carbonate skeletons, becomes more energetically costly for corals. Growth rates slow. The skeletons produced are thinner and less dense, making them more vulnerable to the bioerosion that continuously works against the reef structure. Acidification does not directly dissolve existing coral reefs. The chemistry is more nuanced than that, but it shifts the balance between reef building and reef erosion in ways that are unfavorable to the reef over time. Disease is another significant stressor. Stony coral tissue loss disease, first described from Florida reefs in 2014, causes rapid tissue loss across many reef-building coral species and has spread through the Caribbean with effects that have been described as severe. The disease progresses rapidly across infected colonies, leaving bare white skeleton in its wake, and has affected dozens of coral species across virtually every Caribbean reef system within a few years of its initial description. The combination of multiple simultaneous stressors is particularly significant. A reef facing elevated temperatures, reduced water quality from pollution, reduced grazing pressure from overfishing, and disease pressure simultaneously has less capacity to respond to any one of these challenges than a reef facing only one. Researchers believe that the interaction between stressors, each one making the reef more vulnerable to the others, is one of the most important factors determining which reef systems show resilience and which do not. Interestingly, not all reefs are equally affected by thermal stress. The reefs of the Persian Gulf endure some of the most extreme conditions of any reef system on Earth. Summer temperatures regularly exceeding 35 degrees Celsius, salinity higher than most tropical reefs, and oxygen levels that fluctuate more than most reef-building corals can tolerate. And yet coral reefs persist in the Gulf. The corals there have adapted, whether through natural selection for heat-tolerant individuals or through the adoption of heat-tolerant zooxanthellae strains, to survive conditions that would kill most other reef corals. These Gulf reef corals are of significant scientific interest precisely because of their thermal tolerance. Understanding what makes them able to survive in 35-degree water, when most reef corals bleach and die at 32 or 33 degrees, might provide insights applicable to reef restoration and assisted evolution programs elsewhere. Deep-water refugia are another area of active research. Some reef species exist at depths in the mesophotic zone where temperatures are slightly cooler than at the surface, potentially buffering them from the worst effects of thermal stress events. Whether these deep populations can contribute to the recovery of shallow reefs by producing larvae that drift upward and settle in bleached shallow areas is being studied with increasing urgency as a potential component of reef resilience. The picture is complex. The challenges are real and significant. The science being done in response to them is patient, careful, and ongoing. And alongside the science of understanding reef challenges is a growing science of reef restoration and reef futures. Approaches that are being tested and refined with quiet determination in reef research programs around the world. That is where we are going. In our final section, there is nothing left to explain. The coral polyp has been described. That tiny, tentacled, calcium carbonate secreting animal, barely visible to the eye, that has built, over millions of years, the largest biological structures on Earth. The Zerxanthelle partnership has been described. Two organisms so different from each other, living as one. The alga providing most of the energy that the coral needs to exist. The reef built over deep time has been described. Fringing and barrier and atoll, slow dome coral and fast branching coral, cold water reef in the dark, deep Atlantic. Darwin's century-old prediction confirmed by a drill bit in 1952, reaching volcanic rock 1,400 meters below the surface. The reef at night has been described. The polyp tentacles extended into the dark water, the snapping shrimp crackling audible kilometers away, the bioluminescent organisms glowing faintly in reef caves, the fish larvae following sound through the dark ocean toward a home they have never visited. The mass spawning has been described. The underwater snowstorm of rising eggs, the whale shark arriving to feast on the bounty, the planular larvae drifting for weeks before settling on the reef structure that will be their home for centuries. The hidden interior of the reef has been described. 10 species within for every one visible on the surface. Cave communities of cup corals and sea fans sustained in complete darkness by water flowing through the labyrinthine reef structure. The mesophotic zone below with its distinct and poorly known communities. The social complexity has been described. Cleaning stations and UV communication and goby shrimp partnerships and the pistol shrimp's cavitation bubble reaching temperatures comparable to the solar surface. The predators and the grazers and the invisible engineers have been described. The connected coastal ecosystem of mangrove and seagrass and reef has been described, and the history of how humans came to understand all of this. All of that is there now, somewhere in the mind, held gently, without urgency. What remains is simply to rest in it. The global economic value of coral reefs has been estimated at approximately $375 billion per year through fisheries, tourism, coastal protection, and pharmaceutical development. This number is worth mentioning, not to reduce the reef to an economic calculation, but because it reflects something important about the relationship between human civilization and the reef ecosystem. 500 million people depend on reef fish as a primary protein source. Medicines derived from reef organisms are in clinical use, treating HIV and chronic pain. The white beaches that draw travelers from around the world are, in significant part, made of parrotfish excrement. The coastal communities of the tropical world are shielded from storm waves by coral structures built by animals barely visible to the naked eye. The natural wonders of the reef world are worth naming slowly. The Coral Triangle, the waters of Indonesia, the Philippines, Malaysia, Papua New Guinea, the Solomon Islands, and Timor-Leste, contains approximately 600 species of reef-building coral compared to approximately 70 in the Caribbean. It is considered the global center of marine biodiversity, and its reefs harbor communities of organisms found nowhere else on Earth. The Great Barrier Reef, 2,300 kilometers of living structure visible from space, growing in its current location for approximately 500,000 years, remains the largest biological structure on Earth, a reef system of scale and complexity that has no parallel elsewhere. The Blue Hole in Belize, a circular sinkhole 300 meters across and 125 meters deep, opening in the shallow reef platform of the Belize Barrier Reef, is one of the most geologically and ecologically fascinating dive sites in the world, its walls recording sea level changes from the last ice age in the stalactites that formed when it was a dry cave. Raja Ampat in West Papua, Indonesia, considered by many researchers to have the highest marine biodiversity of any reef system on Earth, supports communities of reef organisms of extraordinary richness in waters of extraordinary clarity. The emerging science of reef restoration is being practiced with quiet determination at research stations and dive operations around the world. Coral gardening, growing coral fragments in underwater nurseries on wire trees or PVC structures, then transplanting them to degraded reef areas, has been shown to be effective in increasing coral cover on damaged reefs and is now practiced at increasing scale. Acoustic enhancement, playing recordings of healthy reef sounds through underwater speakers near degraded areas, has been demonstrated to increase the settlement of larval fish and invertebrates on those areas. Coral cryopreservation, the freezing of coral sperm and larvae in liquid nitrogen for long-term storage, is being developed as a way to preserve the genetic diversity of reef-building species against future loss. Assisted evolution, selectively breeding heat-tolerant coral strains, or transplanting more heat-tolerant zooxanthellae into coral species currently vulnerable to bleaching, is being researched and in early trials. The Persian Gulf corals that survive in 35-degree water are among the models being studied. There are coral colonies alive on reefs today that were growing before European explorers crossed the Pacific Ocean. A porite's coral head on a reef in the Great Barrier Reef or the Indo-Pacific may have been adding its fraction of a millimeter each day for 500 years, growing through the age of exploration, through the Industrial Revolution, through the entire span of recorded modern history, adding to its skeleton the chemical record of every year of ocean conditions it has experienced. Tonight, that coral is doing what it has always done. The polyps are extending their tentacles into the dark water above the reef. The zooxanthellae within its tissue will begin photosynthesizing at first light tomorrow. The tiny fraction of a millimeter of calcium carbonate that it will add to its skeleton tonight is being deposited now, quietly, in the dark, as it has been deposited every night for half a millennium. The reef is working. The snapping shrimp are crackling. The cleaner stations are waiting for morning clients. The spawning night is somewhere in the lunar cycle, approaching or receding. The planular larvae from last year's spawn have found their settlement sites and are beginning, this very night, to lay down the first calcium carbonate of the colonies they will become. The hidden world inside the coral reef is not hidden because it is inaccessible. It is hidden because it requires a certain kind of attention, slow, patient, willing to look beyond the surface, to see. It has been doing its work for hundreds of millions of years. It does not require our attention to continue, but it rewards that attention with something that is difficult to name and easy to recognize, a sense of being in the presence of something extraordinarily old, extraordinarily alive, and extraordinarily worth knowing about. Sleep well.